Okay. I'm gonna show you the first room. The introduction. It's right over here. Here we are.
We received our yeast cultures from the cellular diversity class. Their session 1 started the yeast cultures in a non-selective liquid media. Their session 2 then exposed the yeast cultures to a selective media, and their third session counted the bacterial colonies. Throughout history, there was an argument between Lamarckian evolution theory and Darwinian evolution theory. Lamarckian evolution theory states that changes arise through use and disuse. This supports the post-exposure hypothesis. Darwinian evolution states that random inherited mutations and natural selection are the driving factors for evolution. This supports the pre-exposure hypothesis. Max Delbruck and Salvador Luria were two scientists looking to solve the debate between the evolution theories. After meeting at a conference of the American Physical Society at Philadelphia, they decided to work together. The idea for their experiment arose from Salvador Luria watching a slot machine, and he noticed that the slot machine doesn't behave statistically, and applied this reasoning to bacterial mutations. The experiment took place in the 1940s using E. coli bacteria and bacteriophages. They expected the mean and variance of the colonies to be similar. They concluded that mutations were random and not directed by the mutation, favoring the pre-exposure hypothesis, and thus Darwinian evolution. This experiment marks the birth of bacterial genetics. Rather than using E. coli in our experiment, we used yeast cells, and rather than using bacteriophages, we used cannabinoid. The pre-exposure hypothesis relies on mutations arising randomly before being exposed to a pressure. In this hypothesis, there would be a high frequency of red colonies, creating jackpot plates. The colonies on these plates should all have the same mutation, with different mutations between plates. The post-exposure hypothesis relies on the mutation arising after exposed to a pressure. In this hypothesis, there would be a low frequency of red colonies, and each colony would have a different mutation. The parental yeast strain used in the experiment contained 3 existing mutations. CAN1 encodes a plasma membrane arginine permease. A mutation of CAN1 renders the cell immune to cannabinoid. An AD2 mutation causes red pigment to accumulate in the cells. Sub4O encodes a mutation suppressor of tRNA. If Sub4O has a mutation, then both mutations of CAN1 and AD2 can exist, and the cell is immune to cannabinoid and red. If there's no mutation in Sub4O, then the mutations of AD2 and CAN1 are suppressed, and the cell is no longer immune to cannabinoid.
Let's go to the next room. Here we are. The methods room.
Moving on to methods. The class was put into groups of 4 and each group was assigned a different culture plate of red colonies, A1 through A8, B1 through B8, and C1 through C8. Plates A and B were the jackpot plates, while plates C1 through C8 were individual plates. The individual red colonies were scooped from each of the plates and placed in a corresponding PCR tubes for a total of 24 PCRs. We then lysed the cultures to extract the DNA from the yeast cells. This solution was then centrifuged to form the supernatant and pellet. The supernatant was then pipetted into new PCR tubes. A master mix containing primers specific to the Sub4O mutant DNA sequence was then pipetted in the PCR tubes containing the DNA supernatant. A PCR was conducted to amplify the DNA we obtained. Polymerase chain reaction allows the targeted DNA sequence to be selectively amplified. We then took this PCR product and purified it with a series of buffers. The PCR products that were purified were then pipetted into an agarose gel, and gel electrophoresis was conducted. Gel electrophoresis is when an electric current is run through an agarose gel to separate the DNA into visual bands. We did this to ascertain if there was in fact DNA present from the DNA extraction and amplification that we conducted in the previous week. We successfully extracted the DNA from the A1 through A7 and C3 through C4 plates, and those were sent off for sequencing at our collaborating DNA lab for results.
Okay, we're going to the results. Let's go. Here we are. Come on inside.
Figure 4 depicts electrophoresis of purified DNA. DNA is present in this assay for A1 through A8 at 750 base pairs. DNA is also present for C3 and C4 at 750 base pairs. No DNA is present for B1 through B8, as well as C1, C2, and C5 through C8. Figure 5 illustrates DNA sequences with the accompanying mutations in comparison to the normal SUP4O sequence. Highlighted letters represent where a mutation is seen, except in A7 and A8. In A7 and A8, no mutation is detected because base pairs match with the specific SUP4O sequence. In the rest of the jackpot A samples, a mutation is seen replacing thymine with adenine. At a different mutation point, C3, guanine, is converted into adenine. In C4, adenine is replaced with guanine. The dashes represent identical base pairs as in SUP4O.
Last but not least, let's go to the discussion. Here we are!
The purpose of this experiment was to use the red colonies of S. cerevisiae from the jackpot plates created by the Cell Diversity Lab to sequence the DNA and examine specifically the SUP4O gene for mutation. Depending on the location of the mutation within the SUP4O gene, this would help us determine whether the data supported the pre- or post-exposure hypothesis. In the case of the pre-exposure hypothesis, a greater number of red colonies would be found on a single plate, and the sequencing of the SUP4O genes of multiple colonies found on the same jackpot plate would result in the same mutation at the same location. In the case of the post-exposure hypothesis, there would be a similar number of red colonies across the plates, and the red colonies on the same plates would have the mutations but at different locations. After sequencing jackpot plates A and C, we looked for specific bases to determine where the mutation occurred. At jackpot plate A, samples 1 through 6, a mutation is observed in the same location of the SUP4O gene from a thymine to an adenine. Jackpot plate A, samples 7 and 8 were red, which signified a mutation, but no mutation was found within the SUP4O gene with unknown reasons for this occurrence. Jackpot plate C, samples 3 and 4 were on different plates and contained a mutation of the SUP4O gene, but at different locations. Based on the colonies sequenced from the jackpot plate A dishes 1 through 6, having the mutations at the same locations, this supported the pre-exposure hypothesis because this indicates that the mutation occurred before having contact with the cannabinoid. Jackpot plate C dishes 3 and 4 came from different plates but still had the mutation within the SUP4O gene, which supports the pre-exposure hypothesis by having similar locations of the mutation. In the future, we could sample other red colonies found on C3 and C4 to see if the trend continues similar to jackpot plate A sequences. Further questions can be researched to determine why jackpot plate A7 and A8 were red in color to indicate the mutation but was not observed in the SUP4O gene, which can raise questions as to where the mutation could have occurred to cause the red coloration of these colonies.